Alright guys, today you're going to listen to this video about cell transport, and you're going to use the information on the video to answer the questions that are on the worksheet that the sub should give you. This video is posted on Moodle under the link celltransport2012 if you need to go back over the weekend and listen to it again to help you answer the questions. The first thing we want to mention is the fluid mosaic model of the plasma membrane. Remember that the membrane is made of two layers of phospholipids, phospholipid bilayer. Those phospholipids are said to be antipathic because they have one region that is polar, and those would be the phosphate heads, and one region that is nonpolar, and those would be the fatty acid tails. Even though they're antipathic, the biggest part of the cell membrane is nonpolar, which means nonpolar things can fairly easily cross the cell membrane. Also, very tiny things have no problem crossing the phospholipid portion of the cell membrane. But bigger things, and also ions or other polar molecules, have a hard time going through the phospholipid part of the membrane, and therefore they have to go through structures, proteins, called channel proteins or transport proteins. Because the phospholipid part of the cell membrane only allows certain things to go in and out, sometimes it's referred to as selectively permeable. Or semi-permeable is another word we might use. This allows the cell membrane to perform its main job, which is to regulate what can enter and what can leave the cell. So some factors that affect whether or not a substance can enter or leave the cell are size. Small things typically have no problem going through the phospholipid portion of the membrane. Large things do. Polarity is another factor. Things that are either polar or ionic have a really hard time getting through the phospholipid part of the cell membrane. Things that are nonpolar can pretty much go straight through. Also, certain things have specific channels or transport proteins that bring them in. If those channels are there, they typically go through without much of a problem. If the channels are not there, it's very hard for those substances to get in and out of the cell. Let's differentiate between some of these proteins that are embedded in the phospholipid portion of the cell membrane. Channel proteins are what we've been calling transport proteins. They allow certain substances, sometimes ions, sometimes large things, to move from areas of high concentration to areas of low concentration. The difference here is that these are substances that have to move through a protein. This process that happens with the help of channel proteins is called facilitated diffusion. It's diffusion with a little help. That's what facilitate means. It's a type of passive transport. Things again move from high concentration to low concentration. The difference between this and normal diffusion, which we'll talk about in a second, is that this type of diffusion happens with the help of a channel protein. Carrier proteins, on the other hand, carry out a process called active transport. I want you to think of carrier proteins like pumps. They move materials from areas of low concentration to areas of high concentration. They help you to build up certain materials inside your cells. A good example of active transport in the body is the sodium-potassium pump. It's very important for the formation, or the functioning rather, of your nerves and your muscles. These ions are your body's electrolytes, and they help you to generate electricity for nerve impulses. You also have cell recognition proteins, and these are what we've been calling the marker proteins, or sometimes the name tags. These help to identify your cells to your immune system, so that your immune system doesn't destroy your own cells. And then finally, we talked about receptor proteins. Typically, these are receptors for hormones or some other kind of cell signal. And once the hormone and the signal get together, or the receptor and the hormone get together, that induces some kind of response. It may be, if we're talking about growth hormone, once the hormone and the receptor get together, it causes, obviously, the bones to get longer. But when a hormone and a receptor get together, that causes some type of response inside the cell. It's a type of signal. This slide is just showing you that the phospholipid portion of the membrane is very permeable to small things like water. It's also very permeable to non-polar, non-charged things. But substances that are big, this object here labeled a macromolecule is probably a protein. You can see how it's bent and twisted. It can't get through. And also these things over here, which are ions. This might be like sodium ion. This might be like fluoride ion. Those substances have a hard time getting in and out of your cells. Now, one of the main topics in cell transport is diffusion. Diffusion is the movement of a substance from where there's a lot of the substance to where there's not so much. From high concentration of the substance to low concentration of the substance. When we're talking about diffusion, we often talk about what's called a concentration gradient. This word gradient simply means a difference. So we're talking about a difference in concentration. Diffusion happens, at least net diffusion happens, when there's a difference in concentrations on two sides of a membrane. Sometimes we say diffusion is movement of a substance down a concentration gradient. And that simply means from high concentration to low. Now, we often call diffusion passive transport because the cells that this is happening inside, they don't have to use any of their own ATP or any of their own energy to make it happen. It's powered mostly by the environmental heat found inside a cell or inside a room. So it's due in part to the random, rapid motion of molecules brought about by heat. Sometimes this random, rapid motion is referred to as Brownian motion. And again, that's caused by heat. Anytime there's heat, particles are going to move. But we also need to talk about net movement. Let's say you had a cell, and maybe this line down the middle right here is a membrane. It's a selectively permeable membrane. Let's say these two containers are the same size. We started off with maybe 2 sodium ions over here and 4 over here. Right now there's a concentration gradient. And we know that these sodiums should move from high concentration on this side to low concentration on this side. So they're going to move. But when just one of them moves, now the concentrations are equal. There's no more concentration gradient. There's no more difference in concentration. There's no high and no low. This doesn't mean that diffusion stops. It means that net diffusion stops. Typically what would happen in this situation if one sodium moved over from right to left or left to right, one would probably move back in the other direction so that the balance stays there. Once the concentrations are equal, the system is said to be in equilibrium, in an equilibrium state. And once nature gets things to equilibrium, particles continue to move, but the net movement or the net diffusion will cease. There are several things that can affect how fast diffusion will happen. One of them is the size of the gradient. Whenever there's a really big difference in concentration, diffusion happens very fast. The smaller the differences in concentration, the slower the rates of diffusion. Also temperature. Higher temperatures mean the particles are moving faster, which mean they're going to diffuse faster. The mass of the particles that we're talking about also are important. Bigger particles have more inertia. They're going to move more slowly. They're going to diffuse more slowly than those smaller particles, which have less inertia and are easier to get moving. Diffusion, simple diffusion, is important all over your body. It's how in the lungs, oxygen moves from the alveoli, the small air sacs in your lungs, into the capillaries. The oxygen moves from high concentration in the lungs to low concentration in the capillaries. It's also how carbon dioxide moves out of those capillaries and into the lungs and eventually out of the body. Again, high to low concentration. Another topic that we want to mention here is osmosis. Osmosis is the diffusion of water. And sometimes we'll say it's the diffusion of water through a selectively permeable membrane. And oftentimes, since it's a type of diffusion, it's the movement of water from higher concentration of water to lower concentration of water. But oftentimes that's not the way we talk about it. Oftentimes we talk about it in terms of not the water, but what's in the water. So sometimes we'll say osmosis is the movement of water from areas of less salt or less solutes to areas of more solutes. That's another way, this statement right here, low solute to high solute, is another way of saying this. Because if the concentration of solutes are low, the concentration of water must be high. If the concentration of solutes are high, the concentration of water must be low. So these two statements mean the same thing. Just two different ways of saying the same thing. Water moves from areas of high water concentration to areas of low water concentration, or from areas of low solute concentration to areas of high solute concentration. The two statements mean the same thing. Osmosis is also a passive process. It doesn't require the cell to use its own energy. And in addition, we can define it one other way. But before I do that, osmosis tends to happen when a membrane isn't permeable to the solute that we're talking about, but it is permeable to water. Water can typically go through the cell membrane wherever it needs to, but oftentimes there are also channel proteins that help water go in and out of a cell. And this is a term you should be familiar with. The channel proteins that act as tunnels to allow water to go into a cell are called aquaporids. These are channel proteins that help water to go in and out of a cell, aquaporids. A couple of terms I want to mention: hypertonic, hypotonic, and isotonic. These refer to concentrations. If a solution is referred to as hypertonic, that means the concentration of solutes, dissolved materials in that solution, is higher than whatever you're comparing that solution to. Hyper means higher than, like hyperactive, more active than it should be. Hypoactive solutions have less dissolved solutes in them than what you're comparing them to. The concentration of solute is less than what you're comparing it to. In isotonic solutions, their concentrations of solute are equal to whatever you're comparing them to. So yet a third way of defining osmosis is that osmosis is the movement of water from hypotonic solutions to hypertonic solutions. And again, you should see that this statement is exactly the same thing. It's saying the same thing as these other two. They're all saying that water is moving from where there's a higher concentration of water to a lower concentration of water, or from where there's less solutes, less solute concentration, to where there's more solute concentration. Osmosis is important all through biology. A good example would be if you put plants in really salty water, those plants are going to begin to lose water and they're going to shrivel, because the water is going to move from where there's less concentration of solutes, in this case in the plant, to where there's more concentration of solutes, in this case in the salty water. If you've ever put salt on a slug, you know that the water in the slug is pulled out toward the salt. The water is moving from less concentration in the slug, less concentration of solute, to more concentration of solute outside. But that only happens because the cell's membranes are permeable to water. The water's able to move, but the salt's not. That's when osmosis happens. Osmosis happens when a membrane is permeable to water, but not permeable to the particular solute that we happen to be talking about. Let's talk about organisms living in fresh water. Protists, and also some plants. Protists and plants are typically soaking, at least a lot of them, in fresh water. Fresh water has very few solutes. The bodies or the cells of these protists and plants have more solutes than the water that they're soaking in. Therefore, water continually wants to move into these cells. It wants to move from high concentration of water in the water to low concentration of water inside those protist or plant cells. which means these cells always have a hard time maintaining water balance because water always wants to go into them. To keep from bursting or to keep from having pressure problems, lots of protocells and lots of plant cells have structures called contractile vacuoles. Now we know vacuoles store things, but these vacuoles not only store things, in this case water, they also act as pumps. They help to pump the excess water out of the cells of the protist or the plants. Saltwater fish have a problem with the water too. Saltwater fish are living in water that has more solutes in it than their cells do, which means that saltwater fish are constantly losing water. Water's moving out of their cells into the salty water. That means that to be able to deal with this, saltwater fish have to constantly drink, and they also very seldom urinate because they want to maintain their body water. Freshwater fish have the opposite problem. Their cells have more solutes than the water that they're living in, which means that water is constantly moving into their cells. To get rid of that extra water, freshwater fish typically almost never drink, and they also urinate continuously. They're constantly having to get extra water out of their bodies. Here's an example of what might happen if we soak the plant cell in a hypertonic solution. And again, in this case, hypotonic rather. We soak the plant cell in a hypotonic solution. And remember, what that means is that the solution, here's the wall of the plant cell. The solution out here has less solutes in it than are in the plant, which means that water wants to move from areas of less solute concentration to areas of more solute concentration. Water continually goes into the cell. And because it's going in, pressure starts to build up in the cell. If this were an animal cell, it'd probably burst. But plant cells are protected from bursting because of the presence of the wall. But when plants are put in a hypotonic solution, pressure builds up in them. We say that's called turgor pressure. On the other hand, if you put a plant cell in a hypertonic solution, now there are more solutes out here on the outside of the cell and less solutes inside. And what this means is that the water wants to move out from areas of less solute concentration to areas of more solute concentration. So these cells begin to shrivel. Now notice, the wall doesn't shrivel, but all the inside contents do. This process that happens in plants or any cell that has a wall when it's placed in a hypertonic solution is called plasmolysis. The cell shrinks away and shrivels away from the wall. Similar kinds of things happen to animal cells. Here's an animal cell placed in an isotonic solution. Remember, that means that the concentrations of water and solutes are the same outside the cell as they are inside the cell. Things move both ways. But in this case, there's no net diffusion. There's no net gain of water, or no net gain of water. There's no net loss of water. There's no net diffusion. Diffusion is happening, but there's no net change. Here we've taken this red blood cell and we put it in a hypertonic solution. So now the concentration of solutes are higher on the outside of the cell than they are on the inside of the cell. Assuming that the membrane of this cell is permeable to water, but not permeable to the solute, water's going to move out. It's going to move from areas of less solute to areas of more solute, or areas of higher water concentration inside the cell to areas of lower water concentration outside. The process that we see illustrated in this picture is called crenation. That's when a cell without a wall shrivels because it's placed in a hypertonic solution and therefore loses a lot of water. Over here's the opposite process. Here we take the blood cell and we put it in a hypotonic solution. This means there are less solutes. There's a lower concentration of solute outside the cell and a higher concentration inside the cell. In this case, water wants to move into the cell. Now again, that's of course assuming that the cell membrane is permeable to water, but not permeable to the solute. That's an important point to make. In an animal cell, if this is allowed to continue happening for a long time, this cell is going to swell and swell until eventually it will burst or lyse. There's no wall here to protect this thing, so it just continues to take on water, continues to build up pressure until it pops. Two or three more quick points. Another type of cell transport mechanism is facilitated diffusion. We mentioned this earlier in the video. This is basically diffusion with a little help. This is when substances move from high concentration to low concentration, but they do so through channel proteins. This is one of the main ways that ionic substances, polar substances, and large substances can get in and out of the cell. It's a passive process. Again, the cell doesn't have to use ATP. The energy of environmental thermal energy makes this happen. A couple of other notes about facilitated diffusion. These channel proteins that are involved in the process are very specific. They typically are only going to work for one particular substance. And then one other note. This process is really important for moving in sugars, which can be polar, can also be fairly large, and also amino acids, which can be charged and also polar. This is how they get in and out of cells oftentimes. Another type of transport mechanism we want to mention is active transport. As soon as you see that word active, you should know that this process requires the cell to use energy, requires it to use ATP, requires it to hydrolyze ATP, which means break it down. Active transport is the movement of a substance against the concentration gradient, which means the substances are being moved from low concentration to high concentration. That's not the way nature wants to do things. Nature wants to keep things in balance or to move things toward balance. Here we're going against that. So this requires energy. And the reason you might want to do this is sort of like pumping a substance, building it up in one place and taking it away from another. Probably the most important example of active transport in your body happens at what's called the sodium-potassium pump. This pump pumps sodium out of cells and potassium into cells. Builds up a big concentration gradient. This process, this concentration gradient of sodium and potassium, allows your nerves to generate electricity. It's hugely important for nerve function. Somewhere around 1/3 of all the energy that your body uses is used to power these sodium-potassium pumps. These pumps are proteins. We call the pumps, the proteins involved with facilitated diffusion, channel proteins. The proteins involved with active transport we often refer to as carrier proteins. And they will very often have associated with them an enzyme called ATPase. This enzyme breaks down ATP and thus helps to provide the energy that is necessary to make this process happen. This process is also important during photosynthesis and cell respiration, and we'll talk about those two processes here in the next couple of weeks. The last couple of things we'll mention here involve something called endocytosis. This is the taking in of large particles into the cell. This process too requires the cell to use energy, so it's an active form of transport. In class already, we've talked about phagocytosis. If you break that word apart into its pieces, cyto means cell, phago means eating. So this is when a cell eats. It's when it takes in large solid particles. And it does that by surrounding and engulfing the particle and enclosing it into a membrane. We'll see a picture of how this works in a second. But remember that once that substance is taken in, once it's phagocytized, that it's going to be intracellularly digested. The lysosomes are going to empty enzymes into that vesicle or vacuole that's around the substance, and they're going to break it down. That's called intracellular digestion. It's how your white blood cells get rid of germs, how they break them down. It's how one-celled things like amoebas and parameciums digest their food. Very important process, intracellular digestion. Let's go ahead and look at a picture of phagocytosis and how it works. Notice there's a typo on this slide. This should be a G, not a T. But here we see a cell membrane surrounding this red blood cell. Eventually, this membrane is going to continue to invaginate and pinch until this side of the membrane comes into contact with this side. When that happens, the cell is now enclosed in a vacuole, and the lysosomes will be used to break it down and get rid of it. The last process we'll mention is pinocytosis. This is when a cell engulfs large amounts of dissolved substances, dissolved solids. Could be hormones, could be certain enzymes, could be materials from the blood, could be just simple solutes. So the thing that sets this apart from phagocytosis is engulfing large solid particles. Pinocytosis is engulfing large amounts of dissolved solutes. Sometimes receptors are involved in this process and trigger it to happen. That particular form of pinocytosis, where receptors are needed to trigger the process to start, is called receptor-mediated endocytosis. And I'll end with this. Oftentimes, there are certain viruses, certain pathogens that have learned to use our receptors to get into our cell. And we know that's the case with HIV. HIV has learned to use a receptor involved in this process of pinocytosis as its entry point into some of our cells, and that's a problem. Hope this helps. Remember, this video will be posted on Moodle. You can use it to answer the questions over the weekend. These questions will be due for you guys who are in A classes on Monday and for you guys who are in B classes on Tuesday. Have a good weekend.